Okay, there we go.
All right. Good day, everyone. I'm Cindy Bergeron, and thank you all so much for being here today as we kick off our Spring 2022 series with our first webinar of the year. Today, we have the pleasure of hearing from Dr. Kathleen Benison. Kathleen is from Massachusetts, and she earned her bachelor's in geology and chemistry at Bridgewater State College in Massachusetts. She went on to earn a master's in geology from Binghamton University in New York, followed by a doctorate in geology from the University of Kansas. Dr. Benison served as a professor at Central Michigan University for 15 years before her professorship at West Virginia University, where she is now. Dr. Benison is a GSA fellow, an Explorers Club member, science editor for journal Geology, and a return sample selection participating scientist for the Mars 2020 mission. We are very excited for Dr. Benison's presentation today titled, Why Sample and Return Salt from Mars?
Thank you for that introduction, Cindy. Good morning, good afternoon, good evening, depending upon where you are, and happy International Women's Day. I'm Kathy Benison, and I'm really happy to have this opportunity today to tell you a little bit about why I think it's important to sample salt minerals on Mars and return them to Earth for further study. I am a chemical sedimentologist and an accidental astrobiologist. And so you'll hear about both sedimentology and astrobiology, as well as some planetary geology in this talk. So we know a lot about Mars. We know from orbiters, landers, and rovers' work over the past few decades that there used to be active volcanoes on Mars. There are ice caps on Mars. There are a lot of craters there. There's a lot of evidence that there was once liquid flowing on the surface. And we also have both ancient and modern evidence of winds on Mars. So we know a lot about Mars already. There's a lot of salt minerals on Mars, and in particular, we know about a lot of gypsum. There are whole dune fields that are full of gypsum. This is an outcrop. This picture was taken by the Opportunity rover, and I really like it because we can clearly see three distinctive stratigraphic units. There's a cross-bedded sandstone at the bottom. There is a mud-cracked mudstone in the middle, and then at the top, another cross-bedded sandstone. But the spectroscopy of this outcrop shows that it is composed mostly of calcium sulfate, and it's been interpreted as most likely gypsum. So we have a lot of evidence of different kinds of depositional as well as diagenetic gypsum on Mars. So this is a picture in the upper left of some cross-bedded plastic gypsum. We also have seen veins of gypsum on Mars. And then the picture on the right shows these molds of crystals. And these are pretty similar looking to displacive gypsum crystals that we see on Earth. And on Earth, those displacive gypsum crystals form from growth in shallow saline waters, shallow saline groundwaters, just centimeters below the surface. So these have been interpreted as most likely having once been displacive gypsum that grew from shallow groundwaters and then dissolved. This picture was really exciting to see, and this came down from Mars just last week, and this comes from the Curiosity rover. And people were talking about this on social media. I think this is a splay of bottom growth gypsum crystals that maybe have been abraded by the wind. So this is was pretty exciting to see, because I've seen a lot of things like this when I go out and I study modern gypsum forming in places like Western Australia. But there's also a lot of halite on Mars. Now, halite is much more difficult to detect by spectral means, especially by remote spectroscopy, but we do have evidence of this. We have evidence of it from rovers as well as from spectral data. I should say, spectral data from rovers as well as from orbiters going around the planet. So in this picture, we're looking at this big region where there is the blue color here has been interpreted as chloride salts and most likely halite. When we look closely at these areas that have a lot of halite, they have polygons. And this is reminiscent of some of the halite crusts that we see on Earth in places like Death Valley and Bonneville Salt Flats. Salt minerals on Mars tell us that Mars has a complex history of saline surface waters and saline groundwaters. But the salt on Mars can also help us, sorry about that, let me go back. The salt on Mars can also help us to answer the question about whether life ever existed on Mars. And in order to study and understand Mars salts better, we need to know about salt on Earth. And so these are two photos from some modern acid saline lakes that form salts that I've been studying. And we can do comparative planetary sedimentology by comparing and contrasting the features we see in these terrestrial systems with the rocks on Mars. So on the left, we see a yellow lake, and there are hundreds and maybe thousands of these shallow, small acid saline lakes in Western Australia that are on the Yilgarn Craton of southern Western Australia. So here, the Archean bedrock is very close and sometimes right at the surface, and these lake waters are on top of it. And in this yellow lake, we see that there's white salts along the shoreline and under the lake water. The lake water is yellow here because it has high aluminum. The salts here include halite and gypsum and a few other kinds of salt minerals. And the pH when I took this picture was 2.0. The salinity was about 9 times as salty as seawater. On the right, we see a place called Salar Gorbea that's up in the high Andes on the Chile-Argentina border. And this is a place where it's a very different system from the Western Australia Yilgarn Craton. It has, in Chile, it's on a active volcanic system, subduction zone. It has, it's high up in the mountains. It's at an elevation of about 4,200 meters above sea level, yet the water chemistry is pretty similar. And here we show the blue water was at a pH of 1.8, and the water in the foreground, just above the pinkish gray, is a pH of 1.6. The pinkish gray material in the foreground here is actually microbial mat with a little bit of water sitting on top of it. And so when we compare these systems, we find that the minerals, the sedimentary structures, the diagenetic features are very similar to some rocks on Mars. So these might be really good terrestrial analog systems for some past environments on Mars. So there are lots of different forms of salt minerals. And here we're going to just talk about some early salts. So depositional salts include those that form as cumulate crystals and bottom growth crystals from surface waters. So by surface waters, I'm talking about lakes and lagoons mainly. And the cumulate crystals are the ones that can grow at the at the water-air interface. They can sometimes link together to form rafts that float around like little boats. but they can also form in the water column. And eventually these cumulate crystals sink to the bottom and make beds. But there's also crystals that can grow from the bottom of the water body up. We call these bottom growth crystals. In the case of halite, we call them chevrons. So both cumulate crystals and bottom growth crystals form beds. So they are technically called chemical sediments. But associated with these, we also see some early diagenetic salt minerals, such as displacive crystals and cement crystals that might grow from shallow, early saline groundwaters just below lakes or just below the Earth's surface. So for the purpose of this talk, we're really going to focus on the chemical sediments that form in the lakes and other surface bodies that are formed from cumulate crystals and bottom growth crystals. So here's some pictures of these chemical sediments. So the top row is showing us halite and the bottom row is showing us gypsum. And if we look at the pictures on the left, we see halite and gypsum in situ in both cases, under about 10 centimeters of lake water. In the middle, we see cross-sectional views of those crusts. So in the picture in the middle top, we're looking at a halite crust, and you'll see that the crystals in the middle and bottom are small. Those are cumulate crystals that grew in the water column or at the air-water interface. And then the big crystals at the top, these big blocky crystals, are bottom growth crystals. So they grew at the bottom of the lake. The important thing about these bottom growth crystals in halite is they tell us water depth. They tell us that this was shallow water, less than about 1/2 meter in depth. In the picture below that, we're looking at a gas can core that I made of the gypsum crust and the mud below. So the mud below is made of a lot of hematite as well as gypsum. And then we see these big swallowtail gypsum crystals that are also orange because they include a lot of iron oxides. They also contain some clay. When we look at both the halite and gypsum in the lab, and we take a razor blade and we slice along cleavage planes and we look under the microscope, we see that both of them have growth bands that are full of little fluid inclusions. And these little fluid inclusions are trapped remnants of the lake from which they grew, the lake water from which they grew. So we've actually preserved the parent water in these fluid inclusions in halite and in gypsum. As it grows, halite and gypsum grow really rapidly. And because they grow rapidly, they trap water, the lake water or the other surface water they grow from. They can trap solids. So in the yellow picture in the middle, we see a fluid inclusion in gypsum, and there are 2 yellow native sulfur crystals in there. Air can be trapped. So the bottom middle picture shows a big dark air bubble that's in Permian halite in a primary fluid inclusion, but also solids can be trapped and microorganisms. So insects have been discovered. I've found a lot of insects and insects parts trapped as solid inclusions within halite especially, and microorganisms like algae. There's 2 algal cells here in this fluid inclusion in halite. And if you squint a little and you look really carefully, my cursor is pointing to a very small little sphere, and that, let me see if I can bring that back there. And that little sphere is a prokaryote, either bacteria or archaea. So I want to tell you a little bit about Lake Magic, because this is one of the lakes in Western Australia in which we've done a lot of work. And the first thing to show you is that these are ephemeral and dynamic systems over time. So the first time I went to Lake Magic was in 2005, in the summer of 2005, and there was no salt there. There was water that was salty and acidic, but there were no salt minerals. And we came back 6 months later in January of 2006, and the water looked very different. This lake was smaller, shallower, and it had a greenish color, and it was just full of halite and gypsum. The pH of the water at the time was 2.0. The salinity was 30% total dissolved solid. So just as a reminder, seawater is about 3.5% total dissolved solid. So this is about 9 times saltier. 2 years later, in January of 2008, the lake was totally dry and there was a thick salt crust. We came back a year later and there was no salt at all. There had been recent rains in this area, but the lake water was still fairly acidic and saltier than seawater. And then in January of 2015, I took this picture sitting in the canoe. So this canoe is in all these pictures. I was sitting in the canoe, that's the front of the canoe, and the lake water was this neon yellow color. The pH was 1.8 and the salinity was 32% total dissolved solids. So these, and there was a lot of halite and gypsum forming. So I chose these 4 pictures because they were all taken in January. And even though I've been to this lake in other months, I wanted to demonstrate here that it is not seasons that control these flooding, evapoconcentration, desiccation cycles. It's the local weather. And so you can have a rainstorm that comes through and floods one lake, and another lake 2 kilometers away might stay totally dry or very salty and not get flooded. So Lake Magic, like the other lakes in the area, has low pH, has high salinity, has a complex, unusual chemistry. It undergoes flooding, evapoconcentration, and desiccation. There are fluctuating air and water temperatures. So I've been there at times when the water temperature is 0 degrees C and other times when it's 50 degrees C. There's high solar radiation. And we have analyzed the lake waters to find that there's a diverse microbial community that lives in these acid saline lakes. We can, when we're at the lakes doing field work, we look for signs of life. In the picture on the lower left, these are all from Lake Magic again. The picture on this lower left, I want to call your attention to 2 things. There is a brown stuff along the shoreline and in the shallow water. And this is plant material. It is mainly eucalypt leaves and bark pieces that have been transported down to the lake during flooding by sheet floods. But the other thing here is this yellow foam. And when we look closely at the yellow foam, in places it looks filamentous. When we put some of the water with the yellow foam under the microscope, we see these beaded chains. And this is reminiscent of a lot of fungi. So we wondered if this was fungi. And then when we culture this in the lab, we find out for sure it is fungi. So there's fungi living in this lake. We also analyzed the lake water for bacteria and archaea. So these plots, this slide is really just about those prokaryotes in the lake. Typically, when people think about extreme waters, the microbiologists assume that archaea is going to be very abundant and there's going to be bacteria too, and that might be the only things living in extreme environments. So that is commonly studied in these systems. And sometimes that's all that's studied for the biology. So I want to bring your attention to the lake water and the groundwater here. And these were 2 samples from Lake Magic that were taken within an hour of each other, within about a meter of each other. So pretty close to each other. The thing to notice is the colors. The colors are different and the colors are representing different microorganisms. The light blue that we see so much of in the lake water diagram is class gamma proteobacteria. The purple that we see is abundant in the groundwater is bacilli. And there's some pink, acidobacteriaceae. And then the other thing to notice is there's some gray in both, and those are novel prokaryotes. So there are different microbial communities in the acid saline lake water and the acid saline groundwater, despite their physical proximity. We also tried to do a more comprehensive study of these two samples of lake and groundwater at Lake Magic. So here are the same two samples. The green circles are for the lake water, which was pH 2.3 and 30% total dissolved solids when sampled. The blue circles are for the groundwater that's nearby, that shallow groundwater. And we were really surprised by these findings. What we found is that the lake water had much more abundant and diverse eukaryotes. So the bigger the circle here, the more abundant and diverse the life form. And these eukaryotes in the lake water were mainly fungi and algae. And there's almost no archaea in the lake water, just a tiny amount. And that really surprised us. On the other hand, the groundwater had mostly bacteria. So again, this is showing really two things, that eukaryotes, mainly fungi and algae, are more abundant and more diverse in some of the lakes. And also that different microbial communities exist between the lakes and the close by groundwaters. So we know about microorganisms in the waters, but what about in the salt minerals that are precipitating from those waters? Here are 3 pictures of primary fluid inclusions in halite from Lake Magic. The top photo shows a little bright sphere that's about 2 microns in size here. This is a prokaryote. It's either bacteria or archaea. The bottom picture shows some yellow dimpled balls with some flagellum. This is algae. We think this is a form of Dunaliella algae, which is common in saline systems. And then some of these microorganisms, some of these fluid inclusions contain both algae and prokaryotes, like that little guy. Besides looking in plane transmitted light, we can use UV light to try to better understand the microorganisms and organic compounds. So in plane transmitted light, we see 3 dimpled balls here. And under UV light, they fluoresce blue, and that's characteristic of algae. We also see this little smooth sphere, and we think this is a prokaryote, and we look at it under UV light and it fluoresces green, which is characteristic of bacteria and archaea fluorescence under UV light. And finally, there's this yellow blob that fluoresces pink and purple, which is characteristic of carotenoids like beta carotene. We can more distinctly define the chemistry of some of these organics by looking at them with laser Raman spectroscopy. So this is a fluid inclusion, again, from Lake Magic halite, and it had this bright yellow blob that we suspected was an organic compound. And so we analyzed it with laser Raman spectroscopy, and this is the Raman spectra for that analysis, for this blob. And it shows some peaks that tell us that this indeed is beta carotene. So now let's go to Chile. So this is Salar Gorbea in Chile. And this also makes a lot of gypsum, this water. It also makes some native sulfur and some halite and some other salt minerals. The dark color that you see on top of the salt is wind-blown volcaniclastic material. So there's strong winds in this area. In the surface waters in this salar, we find a lot of salt minerals growing, like I said, especially a lot of gypsum, and that grows bottom growth crystals, we see here in this hand sample. And if we look at those under the microscope, we see that there are dark and light bands. So these are growth bands, and the dark areas are not dark gypsum. They are just where there's a huge abundance of fluid inclusions, and the light is bouncing off all of those fluid inclusion walls and gives the appearance of this being dark. So if we zoomed in on this, we would see something that looks like this bottom right photo, where there are fluid inclusions that have water. They also, these bubbles that we see in these fluid inclusions are hydrogen sulfide. But sometimes those bottom growth crystals get picked up by the strong wind. So this picture in the lower left is a dust devil. It's actually more properly called a gravel devil because it's carrying big pieces of gypsum, and it's white because of that gypsum that it's carrying. And when we look at what it deposits crystals like this one here, where we can see the growth bands. We can see that this was a twinned crystal and it's abraded. It's been abraded by the wind. But the important thing is that if we look on the inside, we see those fluid inclusions that are remnants of the original water that it grew from. So both bottom growth gypsum crystals and their wind reworked abraded crystals preserve primary fluid inclusions. In some of this gypsum from Chile, we find diatoms that are trapped as solids. So we see some pictures of that on the left, and on the right is a UV picture of this that shows some of this fluorescence of the organics in parts of the diatom. So this is fluorescing. And then the picture on the right is showing a really ugly little diatom. So let me walk you through this picture. This is host gypsum. Okay, so we're in a gypsum bottom growth crystal. And there is this big primary fluid inclusion. And within the primary fluid inclusion is liquid, this water. And there's a lot of these little shapes that are algae and bacteria and archaea. And then we have this ugly little shape. This is an abraded diatom. So this isn't a diatom that's been broken by the wind. So it has a story. It must have lived in another water and then got reworked and ended up in the water body that formed this gypsum and then was trapped within this fluid inclusion. So it's telling a story about reworking and recycling by wind and precipitation in this acid saline setting. The fluid inclusions also contain a lot of prokaryotes and some algae and some beta carotene. So in the picture on the upper left, we see a little cockeye, which is a little bright, smooth sphere that is a prokaryote. We also see these kind of rod-shaped things that are microorganisms. There are cells that look like Dunaliella algae, and then there are some fluid inclusions that are just chock full of different kinds of microorganisms and organic compounds. And we've done the laser Raman spectroscopy in this gypsum as well. So the two pictures on the left are shown with combined transmitted light and UV light. So we can see the fluorescence, but we can also see the things that don't fluoresce. So the picture on the left shows this really big primary fluid inclusion. It's got a triangular shape. In it, it has a big gas bubble of hydrogen sulfide. It has some water, but then it has these little blue fluorescent spheres. And we really wanted to analyze those spheres. So we brought this over to our local laser Raman spectrometer, and we analyzed the gypsum because we knew that the gypsum was going to have a signature because gypsum has strong peaks in Raman. And so we analyzed the host gypsum, and that is the blue lower spectra, which confirms that it's gypsum. And then we analyzed this top sphere, and the black spectra that's sitting on top here is from that sphere. And one thing to notice is that spectra for the fluorescent sphere has picked up the peaks for gypsum. So we see the same peaks for gypsum, but then we see some peaks that are not present in gypsum. And that allows us to figure out what these spheres are. And they in fact are some long chain hydrocarbons. So even in gypsum, we can do this laser Raman spectroscopy. Okay, so we now know that extremophiles are in modern surface acid brines, acid brine lakes, and we know that they are entrapped in salt minerals like halite and gypsum that grow from those brines. So then the question becomes, can microbes and organic compounds remain preserved in ancient salts? So this is gypsum, and gypsum does not remain unaltered for long geologic time spans. So we're looking here at Permian gypsum. The photo on the left is an outcrop in Oklahoma, and you can kind of see the bedding. These were bottom growth gypsum crystals, but they have been highly altered, probably through dehydration and hydration and dehydration and more hydration. So as they transition back and forth between gypsum and anhydrite, the fluid inclusions are destroyed. And so they're not very good for doing the kind of work that we've been looking at in modern samples. The picture on your right is also Permian gypsum, and it's bottom growth gypsum. You can see these beds of crystals. And but in this case, this is from a core. It's the same unit, but from a core instead of an outcrop. And the gypsum here has been replaced with halite. So again, it's not preserving the original structures and materials of that gypsum. But there are samples as old as Miocene where it seems like there might be preservation of microorganisms in the gypsum. So this is Messinian gypsum from Northwest Italy, and this is a paper published by Della Pierre et al. In Geology in 2015. And these images are their evidence that there was sulfide oxidizing bacteria that's still preserved within this gypsum. So I think that the oldest gypsum that seems to have a good possibility of preserving biosignatures is Miocene. In contrast, when we look at halite, we can see halite that's hundreds of millions of years old that is preserving microorganisms and organic compounds and their fluid inclusions very well. This is halite that has been only in a certain depth window where it has not been buried so deep that it's going to start to flow or get heated up and altered, but it also has been not right at the surface where it's going to get dissolved by meteoric waters. So here is 270 million year old Permian halite from Kansas. And here's a core of it. We can see it's bedded. We see there's a lot of iron oxides in it. And a lot of those are just trapped as solids within the halite. When we looked at the halite, we see primary fluid inclusions with suspect microorganisms, and we look at them closer and we can pick out structures that look like algal cells, prokaryotic cells, and carotenoids. This is another picture from the very same halite. And here we're looking at solids in the left that are trapped, and they look organic. When we put the UV light on them, there's different shades of blue and green that we see. So we think that those are some microorganisms that have been trapped as solid inclusions within this halite. But there's even older halite that contains good microorganisms. So this is 830 million year old brown formation halite from Western Australia. So the two pictures on the left and the middle, are showing us a chevron or bottom growth crystals where we see alternating growth bands, and we see primary fluid inclusions. The middle one is cumulate crystal of halite. We also see, if we look in photo C on the right, that besides the primaries, so there are patches, cloudy patches marked with P that are the primary fluid inclusions. Besides that, we also see that there is this curved plane of irregular shaped inclusions that's marked by two S's. These are secondary inclusions. So this was a fracture in the halite that healed and made these fluid inclusions, but these formed later not in depositional time. So we really only focus on looking at the primaries because we're interested in the environment and life during the deposition of this halite. When we look closer, we see that some of the primaries, a lot of the primaries have these orangey yellow shapes that are suspiciously look like they might be microorganisms. And when we look up close at an individual fluid inclusion, we see algal cells, we see prokaryotic cells, and then we see organic compounds, which sometimes, like this one, are encircling a gas bubble. In the brown formation, some of the fluorescence is still the same colors that we see in the modern, these blue colors for algae. However, there are other places where we see in transmitted light good evidence, good appearance for algae and bacteria and archaea. But when we look in the UV light, they fluoresce a bright white, which is something we don't see in the modern. And so this has made us wonder if some of these microorganisms have decayed and their chemical composition has changed enough to change their UV response, their fluorescent response. We're not really sure about that though, but we do see the original colors that we see in some modern, the blue in this 830 million year old halite. Another thing that we notice in a lot of the modern and ancient acid saline lake halite and gypsum is are these hairy blobs. And this is a nickname that we've given because we wanted to give an appearance name, a descriptive name, and not a interpretive name. But we think now that these are clusters of microbes in minerals. So in the two pictures in the upper left here, we're looking at how they appear in relatively low magnification in bedded halite. And so you can see these little spikes are what made me call them hairs. But when we look at them under an SEM, we see some morphology such as hollow tubes and spheres that suggest that these are microbial shapes. Under some of these fill a whole fluid inclusion, and under UV light, they fluoresce shades of blue. And laser Raman spectroscopy shows that they have these wide double peaks that are characteristic of disordered graphite. And disordered graphite is the signature that you would get for any kind of microorganism that was once living and then has died. So we think that these are clusters of microbes and little crystals, especially sulfate crystals. So there's one more place to talk about preservation of biosignatures within acid saline lake systems. And these are in hematite concretion. So there are hematite concretions on Mars, but we find them also in modern and ancient acid saline lake systems. The picture on your left is a cross section of a recent outcrop in Western Australia. And there's really two components to the rock. The blue arrow is pointing to reworked gypsum. And this contains fluid inclusions. So these are little grains that have been reworked from original bottom growth crystals, and they contain growth bands with fluid inclusions. We also see these round red shapes, and these are concretions. And in this case, the concretions are composed of hematite and jarosite coatings. And when we look on the inside of them, we find that they commonly have pieces of gypsum and sometimes quartz grains. And this particular one has a abraded halite crystal. So we see this cubic shape, but we can see it's been broken here. And it has a big hairy blob in the middle of it. And so this might be a really great way to preserve microorganisms through geologic time because the organic is trapped in halite, but that halite is protected from waters dissolving it due to the gypsum, due to the hematite and the jarosite coatings around it, but also even thin layers of hematite protect from UV radiation. And so when we think about possible life on other planets like Mars, we think about high UV radiation potentially degrading that organic. And if it's coated with hematite, that's going to protect it from that UV radiation. But I want to also say that even without organics, even if we bring back samples from Mars that are salt minerals and they don't have any organic material, we could still learn a lot from fluid inclusions, because the fluid inclusions in salts can reveal past atmosphere and past water. So we can figure out things like what the water temperature was, what the water composition was, and even what atmosphere was. So where do we look for clues about environment and life on Mars? Well, I think we need to sample and return the salt minerals on Mars. So we know that there are a lot of dune fields made of gypsum on Mars. Those gypsum grains probably originally formed in water and have fluid inclusion. So we should bring some of those back. There are also outcrops on Mars that are sandstones and mudstones that are composed of gypsum grains that again may have fluid inclusions with microorganisms. And there are concretions on Mars, and we don't know what's on the inside of these concretions. We know that they are composed of hematite on the outside, and if we broke them open and looked at the inside, maybe they contain some salt minerals like in acid saline systems on Earth. So for the first time, there is a rover on Mars that has the ability and has been taking samples. This is the Mars 2020 mission's Perseverance rover, and it has a drill rig on it, a little drill rig. There's a lot of other great instruments on the Perseverance rover, but I wanted to show you this. This is the calibration target for Mastcam-Z. Mastcam-Z is one of the really cool cool camera systems on the Perseverance rover. And just out of view on the edge of this calibration target is the inscription that reads, "Are we alone? We came here to look for signs of life and to collect samples of Mars for study on Earth. To those who follow, we wish a safe journey and the joy of discovery." And surrounding the message is the phrase "joy of discovery" in a variety of languages. And this question, "Are we alone?" is really at the heart of the Mars 2020 mission. So the reason to drill samples and bring them back to Earth is to look for signs of life, to answer that question about whether there was ever life on Mars. So the Perseverance rover and the Mars 2020 mission has really 4 goals. The last one, preparing for humans. This is an experiment that was done with an instrument called MOXIE, and it was a successful experiment where the rover sampled bits of Martian CO2 rich atmosphere, and then were able to convert it to oxygen, O2. So that was done successfully. The other 3 goals are really linked together: looking for habitability, seeking biosignatures, and then taking and caching samples. And this is where I think sampling salts is really important. Oh, I should show you one more thing in this picture. This is a selfie taken by the rover. And if you look in the rock in the foreground, there are 2 little holes in it because this is where 2 core samples were taken. This is what the drill bit on the left looks like. And on the right, we see a titanium sample tube. So each little core sample is sealed in one of these tubes. And it looks pretty small here. And this even shows how small these samples are. So these are pictures I took at JPL that show practice cores for this this rover coring sample. So they're the same size as the cores that we're drilling on Mars. And compared to my finger, they look pretty small. They're actually about the same size as an adult index finger. And this may horrify you if you are a field geologist like me, who's used to taking hundreds of pounds of rock and water samples. However, these small samples might still be enough because these are the size of the samples that we use for fluid inclusion work. So I have two samples on my finger here. The big obvious white sample is actually probably a little bit too big to do a lot of our fluid inclusion work, and we would use a razor blade to cut it up. In the blue circle, you can just barely see there's a little chip, and that's the more typical size chip of halite or gypsum that we use for fluid inclusion work. And we know that we can mine a lot of data from tiny salt crystals. So if we're able to retrieve salt crystals from Mars and bring them back to Earth, we may be able to get a lot of great data from samples just as small as that. So in summary, I want to say that any salt minerals in samples returned to Earth should be examined for fluid inclusions. And I think that the search for life on Mars will not be complete without petrographic and geochemical analysis of its salt minerals. Because as Carl Sagan said, somewhere something incredible is waiting to be known. So I want to thank you for your attention, and I also want to acknowledge funding sources here from National Geographic Society, the National Science Foundation, NASA, and Central Michigan University and West Virginia University. I've also been helped with different stages of this work with microbiology collaborators, sedimentology and geochemistry collaborators, and some really wonderful students through the years. Thank you.
Excellent. Thank you so much, Kathleen, for your brilliant talk. And it was so amazing to see all of these samples from Mars and all of the information. And so if you're happy to answer any questions, and anyone has any questions to ask, you can put it on Q&A or either in the chat.
There's an obvious question, I think, which is, have we been finding and sampling salt so far? And the rover sampled its 7th core sample yesterday and has the 8th one coming up soon. And there's actually a little bit of gypsum and a little bit of carbonate and a little bit of halite, we think, in some of those samples.
Well, that's very
exciting. Yeah.
Yeah. And so I have a follow-up question. I'm sorry about my ignorance in terms of the how it works for the rover to take and return the sample. So do you have things that you have like some kind of live information on when the samples are being drilled and how about their composition? Are the samples then will be physically returned to Earth with the rover afterwards, or how is the information that's get, and when do you get that information?
So, okay. So the rover has been working on Mars for a little bit more than a year. It landed on February 18th of 2021, and it's been taking samples, and it's also collecting a lot of scientific data. So we get that data back. The samples, though, will not come back for about another 10 years. So there are plans being made right now to design what they call the fetch rover. And so there'll be a rover and a rocket that land, and the rover will go out and collect the samples that have been taken by Perseverance, put them in the rocket, and then bring them back to Earth. And that makes it sound really simple. It's actually pretty complicated, but that is being worked on right now. And just recently, the team of engineers was assigned and announced for that. And so there's a lot of strategic planning about where do we leave the samples for this fetch rover. And so that's something that we're always evolving our plans for that.
Wow, that's fascinating. And Kathy, so there's one question here from Oscar Fernandez. He asks, is there any way to culture any of the microorganisms in any of these fluid inclusions, or are they mostly dead?
Oh, okay. So I missed a word in there. Is there any way to?
Any way to culture?
Culture them.
Culture.
Yes.
Yeah, sorry.
So other people have done this in Salt on Earth, and there's two ways that they have done this. One is they have crushed halite crystals and then dissolved the halite and then cultured and analyzed with traditional microbiology methods. The problem with that, the limitation of that, is that it doesn't distinguish between primary and secondary fluid inclusions. So it's hard to know the age of those microbes that are cultured, but that's one thing that people have done. Another thing that people have done is used a syringe to syringe out fluid from large fluid inclusions in halite and culture it and identify at the species and genus level again. The problem with that is that the largest fluid inclusions tend to not be primary. So neither one of those methods is perfectly ideal, but that has been done. And so I imagine when samples come back from Mars, if there are salts, we probably would start with the type of work that I showed today, the petrography, some of the chemical work like laser Raman spectroscopy, and then probably microbiologists would then take those samples and try to culture them. Another kind of part of that question is about, are these things dead or alive? And that's a tricky question. We think that some might be alive because they have what they need in the fluid inclusions. They have nutrients. We see, especially where there's both prokaryotes and algae, we also see organic compounds that might be showing some evidence of microbial activity, but some of them may be dead. And also some of them may not have been living in the system. Some might have been blown into the systems. So I think a lot of the microbial populations in the modern lakes are a combination of things that are living in the lake and then things that have blown in.
So we have one more question from Sean Brennan. What are the plans to keep the samples warm on the return trip? Will the low temperatures affect the samples?
That's a really good question. And there, I was just on a committee that was trying to address that question. And our concern wasn't about them being cold. Our big concern was about them getting overheated. And what we realized is that gypsum was a bit more sensitive to that probably than halite. So these little tubes are going to do a good job insulating, but we think that the temperature range may get as hot as 60 degrees Celsius at times. And I think there's been more concern about that than about the cold temperatures.
Thank you. Yeah, so those are the questions we have so far. Is there anyone else who wants to ask?
Yes, I'm Nawaz. Hello. I have a question. Yeah, I have a question. in terms of biological development, if the planet would have been conducive to life at present, where the life forms would have developed in terms of biology?
Yeah, that's a good question. And people have hypothesized that on Mars, early life forms may have formed in hot springs, in volcanic areas. Some people have proposed that they're forming below the surface. So I don't think we can guess about that. And we basically look to Earth for what we know about early life on Earth. So we don't have any good answers about that. But, I think that Mars is cold right now, but there's a chance that there's permafrost on Mars that melts occasionally when it does get warm. And, so there may, and we know that there's some ice on Mars. So there are places where potentially there could be life today on Mars as well. So we have there's a lot of questions, a lot less left to learn about that.
Okay, thank you.
Thank you.
So if anyone else has any questions, please feel free to put in the Q&A. And also, just before I forget, I'm copying in the chat a link for a survey that South Basins group are doing, because we are planning to do a salt field work related to salt in Portugal later this year. So we would like to gather some opinions from salt tectonics community. So if you'd like to answer the survey, please, if you are interested in this potential field work. So you can find the link in the chat. And is there any other questions for Kathleen?
Thank you, everybody, for
Yeah.
your attention. Thank you for inviting me.
Thank you so much, Kathleen. It was such a pleasure to have you here today.
Yeah, thank you very much.
Have a
Kathleen.
good day or good night, everybody.
Yes.
We learned a
Thank you,
lot today.
Kathy. Okay, goodbye,
you.
everyone.
Goodbye. Thank you.